Good morning, student. I am Dr. Gnanasekar, Department of Medical Electronics, Savitha Engineering College. Today I am going to discuss about the topic Auto Analyzer. Auto Analyzer system. A auto analyzer sequentially measures blood, displays this on a graphical readout. This accomplished by mixing, reagent, reaction, and colorimetric measurement in a continuous stream. So this is a system, auto analyzer system, where the sample is kept in a tube which has been arranged about n numbers, maybe for about 100 or 1000 depending on the size of the unit. And you can see the mixing of proportionality pump and a modifier. So here two containers are there. In first container it will be a sampler, in other container it will be a reagent. And you can see the propagation movement where the dark, that is the spiral tube is there, where the dark is nothing but the sample, next is your air bubble, and third one is nothing but the reagent, and which is going to be added. Next is your dialyzer unit, maintaining the filtration and everything, and heat bathing, maintaining the temperature, and colorimetry, where it finds the absorption technique and the transmission technique, so that what sample it is, and correspondingly the reading out. So we can see the clear system information in detail. Sampler aspirates sample, standards, and wash solutions to the auto analyzer system. Proportionality pump and manifold introduces mixers, sample with reagent to effect the proper chemical reaction to be read by the colorimeter. It also pumps fluids at precise flow rate to other module as the proper color department, that is development, depends on reaction time and temperature. Dialyzer separates interfacing substance from the sample material by permitting selective passage of sample components through a semi-permeable membrane. Heating part heats fluid continuously to exact temperature, typically 37 degrees C, incubation equivalent temperature. Temperature is crucial to color development. Colorimeter monitors the changes in optical density of the fluid stream flowing through tubular flow cell. Color intensities, optical densities proportional to substance concentration are converted to equivalent electrical voltages. Recorder converts optical density electrical signal from the colorimeter into a graphical display on a moving chart. The heart of the auto analyzer system is the proportionality pump. This consists of a peristaltic, accumulating or roller pump. Air segmentation in mixing tube separates the sample reagent mixture from the cleaning fluid and other samples. So no samples will be interrelated. As these air separate fluids transfers the coil of the mixing tube, effective mixing reaction is achieved. One problem with automatic analyzer is contained identification of samples. Patient data can be intermixed with that of other patient if care is not taken. Sterilization is also needed for samples, glassware and equipment parts that are contaminated with disease. Diseases such as hepatitis or other communicable infections can be spread to equipment operators. Maintenance, frequent calibration adjustment. Most problems are mechanical tubes, moving pump parts, and electrical switches and motors. Electronic failures are few. Operation and service manual must always be consulted. Thank you.